color. And here, so it was actually present on the genes responsible for this um, character are present on X chromosomes, and Y doesn't contain this character. Okay. Therefore, so mostly on females. So they have, means because all the genes are present on X chromosome. So in females, at least if one X chromosome is having a normal gene, so even then they can actually they don't express this disease. But in males, there is only one X chromosome. Suppose if the genes which are responsible for this disease are present on X chromosome of males, then definitely they will get expressed. Okay, because there is no other chromosome for compensating the genes. Okay. So the retinas of man's eyes contains certain sensitive cells which are necessary for the distinguishing of red and green color. So therefore, the genes controlling this defect is recessive to the one controlling the normal vision. So normal vision, so the genes responsible or the allele responsible for normal vision are dominant, and the allele responsible for color blindness are recessive. So the heterozygous female looks normal because heterozygous means that is one chromosome is having dominant allele and the other chromosome is having recessive allele. So dominant allele will give normal vision. Recessive allele will give color blindness. So as there are two copies, so definitely the dominant will get expressed. Therefore, the female will be normal. Whereas in males, they have only one chromosome. X1 X chromosome. So if the dominant one is present, that's okay. If recessive allele is present, definitely that will be expressed in males. Okay. And these females, although they express normal vision, but they have this recessive allele, right? So that can be passed to the next generation. So that is called as, therefore the woman, the female candidate here is called as carrier, because they have the allele, but they are not expressing. Okay. So the inheritance of this genetic disease can be studied in the following types of marriages, where normal female and colorblind male. So therefore, normal female, see, here you can see the normal female means they can be homozygous or heterozygous. Okay. And colorblind male means definitely they have defective X chromosome. Okay. So what happens? So the X from male will be inherited and one X from female will be inherited in the child, female child. Okay. So if normal female is dominant, they have both dominant alleles, definitely there is a dominant allele. So other allele, even though it is defective, they will be normal. And definitely the male won't get, because normal allele will be from mother and Y chromosome, definitely it is not having the G. Then in the colorblind female and normal male, so the colorblind female and normal male produce color-blinded sons because X chromosome comes from mother and the sons will have only one X chromosome. Therefore, definitely they will be colorblind and normal vision daughters. So because one chromosome from the X chromosome from the father, so which will be dominant obviously if he is normal vision. So therefore that will express and the daughters will have normal vision. Then carrier female and colorblind male. So colorblind male also is having X chromosome and carrier female also will have one defective allele. So if both defective alleles arise, then the daughters will have the chance of colorblind. Okay. And even then, if a dominant allele is coming from the mother, they might be normal vision also. And the son will have definitely normal vision, 50% of the sons, and 50% of the sons may be color blind. Are you getting my point? That is, now here man is color blind. So definitely one X is coming from the father. If another X is coming from the X, that is defective X is coming from the mother, the daughter will have the color blind. If normal vision allele is coming to the daughter, she will be having normal vision. Same way, male, he is getting Y chromosome from the father, so there is no chance of color blindness from father side. From mother side, if as she is carrier, she is heterozygous. If she is passing normal allele, then the son will be normal. If defective allele is passed, then he will be colorblind. Okay. And carrier female and normal male. So here also we can see color-blinded males and carrier females. You can just make a cross and you can practice and you will understand. Okay. So from the above crosses, we can derive the fact that color blindness is common in males than in females because they have only one X chromosome and the chance of expression will be more. This is because one gene in males and two genes in females are necessary to produce this defect. And all females are carrier and males are never carrier. Either they will be normal vision males or colorblind males. And women with normal color vision but who had color blinded father produce normal and colorblind son in the same ratio, that is 50 is to 50. Okay, that is 1 is to 1 ratio. And color blinded women always have color blinded father and always produce color blinded sons. Okay. And colorblind women produce colorblind daughters only when their husbands are also colorblind. And when both the husband and wife are colorblind, all children carry this defect. Okay, so these are the inferences. Then hemophilia. So hemophilia is nothing but the bleeder's disease. So where it cannot stop bleeding. Okay, so it's a genetic disorder found in man and they lack clotting factor in blood. And if there is any injury, it will lead to continuous bleeding, which is very fatal. So it's called bleeder's disease. So it is also like color blindness. So it is specific to males. And there is one Alex. He was a prince of some country, at France or something, some royal family. Actually, he had that disease. So actually, because though they took a lot of precautions that he was, as he is prince, so he was taken care that there were no injuries and everything. Because at that time, even now there is no particular treatment for that. But at that point of time, they don't even know what the disease is. Then what happened during some war, actually the opposite people, the enemies, they came to know that he has such disease and actually they made some cut on his body and because of continuous bleeding he died. So because of that, the complete empire, the kingdom has lost the war. So that means, see, a single genetic disease has led to the loss of a kingdom. Okay, so that was the history behind that. Then actually the research has done and everything. So then they came to know that hemophilia disease. So then heterogametic female and homogametic male type. So this I told you already yesterday. So ZW, ZO, again I'm not discussing. Then dominant sex-linked genes. So sex-linked genes for white eye color in Drosophila and color blindness in man described earlier were recessive characters. So that means when they are expressed in recessive form. Okay. Some dominant genes are also present in man. So especially genes for defective tooth enamel. Okay. So that was actually dominant sex-linked gene. And this is dominant. So it expresses even in the heterozygous condition. Whereas in color blindness, if they are present in the heterozygous, they won't express. So they will be in the carrier form. But here, as they are dominant, even the heterozygous condition, they will express. And naturally, women have twice as many chances of men receiving this gene. Therefore, women possesses this defect almost twice as frequently than men. In case of recessive genes, men possess the defect twice, almost twice, as frequent as women. Okay. Then sex-linked genes. So the type of autosomal genes which can express in one sex and cannot express in other sex because of difference of their hormonal environment or anatomical dissimilarities, they are called as sex-limited genes. Sorry, not sex-linked, sex-limited genes. So the best example of sex-limited genes is plumage pattern or feathering in domestic fowl of Leghorn breed. So here, males have long, pointed, curved, fringed feathers on tail and neck. But feathers of females are shorter, rounded, straight, and without fringe. Thus, males are cock-feathered and females are hen-feathered. Okay? And by crosses, the kin has been shown that hen feathering results from the single gene H and the cock feathering results from the another allele, small h. Okay. So therefore, and this is depending on the sex hormones. Like as we have like estrogen and progesterone from females and testosterone for males. So in the same way, the hormones in the cock and hen, so they actually influence the expression of this gene. Okay. So this is one kind of sex limited gene. That means it is only limited to that particular sex of an individual. Then sex influenced gene. That is the type of autosomal or sex-linked genes whose phenotypic expression will be either as dominant if the individual is male or as recessive if the individual is female. So these are called sex-influenced genes. That means the gene will be same, but sex will influence its expression. It is baldness in male. So in man, so baldness is seen mostly in the man, that is males, but not in females, right? So in 20s or early 30s, they slowly become thin and the crown of the head, leaving finally a fringe of hairs low on peripheral part of the head. And this is called pattern baldness. And the gene responsible for this is S gene. So it's dominant in males and recessive in females. That's why baldness we see in males mostly. Even in females, very rarely we see baldness. Okay, so these are all the different kinds of patterns. That is Mendelian inheritance, non-Mendelian inheritance, and sex-linked genes, all these things. Okay. So now let us go to the next topic. That is mutations. Okay, just a second. Okay. So now we discuss about mutations. So mutation is nothing but a permanent alteration and it's a sudden change which is seen in the nucleotide sequence of the genome of an organism. So it's a sudden change means it is not present from the birth. Okay, due to exposure to some radiation or some chemicals or such kind of things or due to environmental factors. So suddenly the nucleic acid or due to the accumulation of errors during DNA replication, especially during meiosis and due to the damage of DNA, so it can undergo some mutation. So usually during DNA replication, there is a repair system which will try to repair the errors, but sometimes it will miss. So when it is missed, then it will continuously starts and it accumulates. So here there can be some insertion of sequence or some deletion of sequence, and sometimes this mutations can express in the form of phenotype. Okay, so this results in normal and abnormal processes. That is, during evolution, actually this mutation is good, so that it will give some variation in the organisms, but it can also cause some diseases like cancer and some immune related. So because even during immune system development also there is a chance of this variability at all, like in the antibody formation and all these things. So sometimes it's good and sometimes it is bad. And the genomes of RNA viruses are based on RNA rather than DNA, and they can be double stranded or single stranded. And in some viruses the replication process, because there is no mechanism, so it will take the help of the host to replicate. This is as there is no checkpoint, because if it is the host genome, so suppose human beings genome, so the genome will check its DNA replication and all this. But as virus is invading, so there is less chance of what you call checking, okay, checkpoint of error. So it is very error prone mechanism, and this results in mutations. And mutation in genes can also have no effect. Sometimes there will be no effect, no positive effect, no negative effect. Okay. So one study on genetic variations between different species of Drosophila suggests if a mutation changes a protein produced by a gene, the result is likely harmful. Okay. That is, suppose there should be some kind of change. So the what is the DNA, like what is the final step of having DNA? That is, DNA will give rise to RNA, RNA will give rise to protein, and that protein can be a structural protein or function. They will have some function in terms of like whether they can be secretory, they are receptor or so. So if this DNA, there are mutations in DNA, and that mutations are reflecting in the protein sequence or protein function, then it will be harmful. Okay. And these mutations can be classified into two types: chromosomal mutations or chromosomal aberrations, or gene mutations or point mutations. So what are chromosomal mutations? So chromosomal mutation is nothing but changes that occur in the structure of chromosomes during cell division. So here the chromosomes may break, or chromosomes may join with other chromosomes. Okay, some chromosome may be lost, some part. So the deletion, insertion, or breaks. So any kind of a duplication, so any kind of such changes are nothing but the chromosomal mutations. And inversions are also can occur. That is, the chromosome will like invert. So in the reverse direction, that will come and attach. So first it will break, it will get in reverse orientation, and again it will join. So, and mutations are located on either sex chromosomes or autosomes. If they are on sex chromosomes, that can be inherited. If they are on autosomes, the inheritance chance is less. So they are broadly categorized into variation in chromosome number, variation in chromosomes, arrangement of genes on the chromosomes. First, chromosome number. So haploidy. Haploidy means actually if 2x is a diploid, so haploid is only one set of chromosomes are present. So in corn plant, 20 pairs of chromosomes are present, but haploids with only 20 chromosomes, they appear. But in animals, we don't see this haploidy. Only in the bees and wasps males, we can see this haploid, while females are diploid. And the haploids are smaller than the normal diploids. Okay, so haploid is usually seen in the plant breeds. Then polyploid. Polyploid means increment of the number of chromosomes. Usually there will be diploid number, but multiples of haploid numbers, that is into X. Suppose triploid, that is 3X, tetraploidy, that is 4X, pentaploidy, hexaploidy, octaploidy. Such kind of increased number of chromosomes are also seen. So in plant varieties, we can see like pears, blueberries, blackberries, cherries, cranberries, wheat, grapes, apples, etc. are polyploid. But it is also in animals, it is very rare. However, treating eggs that produce triploid Drosophila and rabbits. So before fertilization, if we treat it with the chemicals, there is a chance of polyploidy. And even in man also, very rarely there are a highly abnormal boy was found to have 69 chromosomes instead of normal diploid number. So this was very like obvious case of triploid. His chromosome complement was XXY. He was a male with abnormal cerebral development and syndactyl of hands and feet. That is fused, fingers will be fused. And the individual survived only fetal life and died at birth. Okay, because obviously there is a very less chance of survival. Then heteroploidy. So it is the change in the chromosome number of a single haploid set of chromosomes. So such changes are brought by phenomenon of non-disjunction discovery by bridges in Drosophila. So non-disjunction means it is a failure of the separation of the chromosomes. So non-disjunction is the failure of two homologous chromosomes to segregate during cell division. So whenever this occurs in somatic cells, two types of daughter cells are produced. One is having the extra number of chromosomes and the other daughter is having no chromosomes, like less number obviously. So because see each cell should have two copies. So if one copy is going to the other cell, so one cell will have three copies, other cell will have only one copy, such kind. Okay, so non-distinction of sex chromosomes may give rise to XXY sex chromosome complement in man. They usually suffer from Klinefelter syndrome. Such syndrome is outwardly man, but they are sterile due to underdeveloped testes and show tendency towards a female breast. So they will express some female sexual characteristics, and though they are males, but they are sterile. Okay, they cannot give birth to the offspring. So there are also XXX female. They are underdeveloped reproductive organs, and second is sexual characteristics also very underdeveloped, but they may be fertile. Okay, they are normal. Though they appear normal, but they have the reproductive organs are underdeveloped. So, but they can give birth. So, with the medical supervision. So, variation in number and arrangement of genes within a chromosome. So when aberrations remain confined to a single chromosome of homologous pair with variations in number of genes and their arrangement, okay, that is described as intrachromosomal aberrations. So within the chromosome only there will be changes. That is deletion, duplication, inversion and translocation. So deletion is nothing but, or deficiency is nothing but, so here you can see that some part will be missing. So that part is having some gene or genes. So this deletion can result in the removal of some set of genes. Okay. A deletion may be terminal or interstitial, that is at the ends or in the middle of the chromosome. Okay. So the account for the notched condition of wing margin in some fruit flies. So Bridges and Moore in 1945, they suggested loss or deletion of a small segment in the X chromosome. And this is that homozygous deletions are lethal. If they are deleted on both the chromosomes, then it will be very lethal. And these deletions can be induced by ultraviolet and X-rays. So depending on the location where the deletions are there, they are terminal and interstitial. Terminal means at the ends. Okay, again it may be two types. So it can be occur in one chromosome which is heterozygous deletion, or on the two sets of chromosomes, so which is homozygous condition. Okay, homozygous deletion. Then interstitial deletion is nothing but in the intermediate position or between telomere and centromere. Okay, such loss of intercalary portion of chromosome is known as interstitial or intercalary deletion. Okay, so in both the cases, the deletion portion may contain one, two, or several genes. Therefore, the deletion of this chromosome results in the loss of some genes. Okay, some may actually reunion. Sometimes the breaked portion may reunite with the original sequence, but it is very, very rare. So what are the genetic effects here of deletions? They affect fertility, viability, crossing over, phenotype, and karyotype. So what is fertility? So the pollen fertility is reduced by the presence of deletion. So especially in plants here, the pollen produced by the deficient heterozygous. So there are two types, functional and non-functional. So the pollens with normal chromosomes are functional and the pollens with deficient chromosomes are aborted or non-functional. Then viability, that is the survival. So very small deletion in heterozygous state is viable, but if it is present in homozygous condition, it will be lethal. And the crossing over. So the crossing over is suppressed in the region of deficiency due to lack of corresponding segment. Then phenotype. Deletion also affects the phenotype. So in the absence of this dominant gene in the deleted region, so the recessive gene expresses even in the single dose. So the example is in man, a syndrome called cat cry. So this results in the deletion of short arm of chromosome. So babies with this defect, they cry like a cat. Okay. Then karyotype. So karyotype is nothing but the mapping of the chromosomes. So generally the chromosomes with deletion can never revert to the normal condition. Okay. So its significance is that the deletion play an important role in species formation and releasing variability through chromosomal mutations. Deletions are important cytological tools for mapping genes, and deletion mapping is used in Drosophila to locate various genes on the chromosomes. Okay. The next is duplication. Duplication is nothing but a small segment of the chromosome will be repeated or doubling or adding of a chromosomal segment. So this extra chromosomal segment may be a free fragment with a centromere or a chromosomal segment with non-complement. They will form like a loop in the homologous region. So again, there are translocations, inversions, rings, eccentric or dicentric chromatids. So they are nothing but the different chromosomal aberrations. So Morgan and Sturtevant discovered this phenomenon of duplication in 1923, and this was detected in bar-eyed mutant in Drosophila. Okay, so here you can see. So wild type, we have that black colored one gene, and this is with strips, and that is female, and the male is having vermilon. So in the below you can see, so the vermilon is having, so one segment is missing here. Okay, so it is shifted to the male. Can you see that? So here there are different types of duplications: tandem, reverse tandem, displaced, and reverse displaced. Tandem is nothing but the sequence of genes in a duplicated segment similar to the sequence of genes in the original chromosomes. So same, like here you can see A, B, C, B, C, like that. If same way it will be present, A, B, C, A, B, C, like that it will repeat. Whereas in reverse tandem, that will be, the duplicate segment will be in the reverse direction. A, B, C, C, B, A, like that, reverse direction. And the tandem and reverse tandem duplications are known as adjacent duplications, as they are adjacent to the original segment. Then displaced. So when the duplication is found away from the original segment, but on the same of the chromosome, that is called displaced duplication. And reverse displace means they are present away from the original segment, but on the other arm of the chromosome. So usually there will be P arm and Q arm. So that is, if from one arm it is dislocating to other arm, then that is called as non-adjacent duplication. So origin, from where does, how they originated means, it is believed that duplications originate due to unequal crossing over during meiosis, and the homologous chromosomes usually they pair in such a way that the identical loci match with each other in their positions. Okay, and this facilitates equal crossing over between non-sister chromatids, and sometimes homologous chromosomes pair in such a misaligned manner that the corresponding identical loci do not fall opposite to each other. Okay, such situation lead to unequal crossing over between non-sister chromatids. This gives rise to two types of chromatids, one with the duplication and other with the deletion. And when a gamete with duplication unites with the normal ovum, leads to the formation of a zygote with triple genes, because one is duplicated, right, in a particular segment of the chromosome. Then genetic effect. So duplications have been found to affect the phenotype, crossing over, gene number, and pollen fertility. So phenotype, duplication of certain genetic regions produce phenotypic effect, like bar I in Drosophila. And crossing over is also suppressed in duplicated region because lack of corresponding duplicated segment. And the gene number is increased in duplication, and the presence of duplication will reduce pollen fertility in plant species. And its significance is nothing but the duplications are less harmful than the deletions and do not reduce the viability of the individual, but they may lead to addition of some genes in population which after mutation play an important role in the evolution. And moreover, duplication sometimes cover the deleterious effects of the deletion in the individual. Then comes the inversion. So inversion is nothing but intrachromosomal aberration where the segment is inverted into 180 degrees. Like here you can see 1, 2, 3, 4, 5, 6. That breaks between the 2, 3 and 5, 6. Now it will be 1, 2, 5, 4 and 3, 6. That means the segments are inverted. And these are two types, pericentric and paracentric. So pericentric is nothing but when the inverted segment of chromosome contains centromere, they are called as pericentric inversions. And if crossing over occurs within the loop of the pericentric inversions, they result in the chromatids including half of the chromatids with duplications and deficiencies, forming non-function in the half of the chromatids. Whereas in the paracentric inversions, when the inverted segment includes no centromere and the centromere remains located outside the segment, that is called as paracentric inversions. So they affect fertility, crossing over, gene order and karyotype. So the crossing over in the inversion loop, they leads to the formation of chromosomes with duplications and deficiencies. So 50% of the individuals they become sterile. And crossing over, so where the inversions, they have pairing problems because that is inverted. So crossing over will be reduced. And gene order, so the gene order will be definitely changed because it is inverted. And the karyotype, so pericentric inversion sometimes result in the change of karyotype by shifting the position of the centromere. Okay, and significance is inversions are very useful in production of duplications and deficiencies, and a large crossover in the inversion loop produces chromatids with duplication and deficiency. And inversions also play an important role in the evolution of new species by changing the karyotype of the individual. So these inversions, they suppress the crossing over and tend to remain in the original combination of genes. And they help in preserving original gene combinations. And inversions provide strong evidence that out of 4, only 2 chromatids are involved in the crossing over. Okay. Then translocation. It is nothing but the reciprocal translocation where the interchange of segments between non-homologous chromosomes. So there are 2 types: homozygotic translocation, heterozygotic translocation. So in homozygotic translocation, normal meiosis occur and cannot be detected cytologically. We cannot see that. And genetically, they are marked by altered linkage group, by the fact that the gene with new neighbors may produce different effect in the new location. And heterozygotic translocation is a considerable degree of meiotic irregularity that occurs, and during meiosis, an individual who is heterozygous for reciprocal translocation, they form cross-shaped configuration in order for which affect the pairing of homologous segments. So this was first detected by Bridges in 1923 in primrose and maize. Okay, and this may be possible through radiations. And simple translocation is nothing but when a segment from one chromosome is transferred and attached to the end of the non-homologous chromosomes, it is called simple translocation. Then shifts. Here the transfer of intercalary segment, that is the central segment, that is interstitial. Okay. In the non-homologous chromosome, it's called as shift. And reciprocal translocations is nothing but where the mutual exchange of segments between non-homologous chromosomes occur. It is called as reciprocal translocation. So in case of reciprocal translocation, the homozygotes on both chromosomes of 2 pairs are involved, and the origin here of translocations is mainly due to breakage and exchange of parts between the non-homologous chromosomes. And when one chromosome from each pair of 2 homologs are involved, it gives rise to translocation of heterozygous. And when both the chromosomes are involved, it is called translocation homozygotes. So it mainly affects the sterility, crossing over, karyotype and phenotype. So sterility is translocations lead to duplication and deletion of genes. Therefore, the gametes with duplication or deficiencies are inviable, and 50% of them will be sterile. Then crossing over, they generally are suppressed in translocated chromosomes. And karyotyping, so the translocation brings changes in the number of chromosomes. Therefore, karyotype also will be altered. And it can also change the phenotype and results in Down syndrome, which is called Mongolism in man. So here there are 3 sets of 21 chromosome. Chromosome 20, first chromosome are present. So the significance here is the translocation alter the chromosome size, chromosome number and karyotype. Thus play important role in the formation of species. And it can also produce new linkage relationship. And these are useful in locating the positioning of genes. Okay. And the centromeres and other genetic markers on the chromosome. So they are helpful in the breeding. Then aneuploidy and polyploidy. So I will just explain you this table. So aneuploidy is nothing but it is having 1 or more extra chromosomes or 1 or more few chromosomes. So hypoploidy or hyperploidy. Hypoploidy means which will reduce the number of chromosomes. In the monosomy, 1 chromosome will be less than the original one. Nullisomy means both will be absent. And hyperploidy means trisomy, that is 3 copies will be present, and tetrasomy means 2 extra copies, so 4 chromosomes. Euploidy, again I told you already, euploidy, so where there are monoploidy, diploidy and polyploidy. So in the aneuploidy, so the as I told you, non-disjunction usually due to the inability to separate segregation. So they leads to various genetic disorders like Turner syndrome, uh, Klinefelter syndrome. So these are important topics for your small questions, like okay, short questions. So here you have 44XO chromosomes. So in females, usually we see, so they are sterile, short stature, web of skin between the neck and shoulder. This suffers a look which is different from the normal females. Okay, even they will have some other heart defects and some other defects also, and there are no cure here. So that is here what is happening, one extra chromosome is present. Then Klinefelter syndrome, where it has 47 XXY or XXY syndrome, it is also called as. So one extra X chromosome is present in the males. Okay. So it is also very rare, but the primary features are lanky body, sterility, small testicles, then less body hair, breast growth. So like they are males, but they appear females in some secondary sexual characteristics, and the intelligence is usually normal. Reading and learning difficulties are present, and the problems in speech are also very common. Then Down syndrome. So this is mainly the non-disjunction of autosomes, but not allosomes. So here also 47. So 44XY and 44XX. So the autosomes, like they have trisomy of 21st chromosome. So affected person has mental retardation, if broad, flat face, eyes with an epicanthic fold, short stature, short hands. Okay, all these are the symptoms. Females are fertile, but they may produce normal or trisomy progeny. And males are sterile with few exceptions. And three kinds of regular trisomy are seen. All cells with extra 21st chromosomes, translocation, where extra 21st chromosome attaches to another chromosome, and 4% are we can see with such kind of condition. And mosaic, some cells have additional chromosome 21. Okay. Then Edward syndrome, where trisomy of 18th chromosome. Okay. Then you have Patau syndrome, where trisomy of 13th chromosome. Then polyploidy. Okay. So anyway, I'm not going to discuss all the examples. Okay. All are important only. Okay. Then polyploidy, the chromosomal mutation in which the cells have extra sets of chromosomes. So instead of diploid, they are triploid or tetraploid like this. So the polyploidy is a state of cell or organism having more than two paired sets of chromosomes. So in addition to the polyploidy, some tissues of animals have diploid, such as human muscle cells. They are called endopolyploid. And species whose cells do not have nuclei, like the prokaryotes, they may also exhibit polyploidy, like especially in the bacteria. Epilopium fischeloni. So we can see in that also. Then in flowering plants like Hibiscus rosa sinensis, in Brassica genus, we see tetraploidy. Okay, Xenopus, that is frog. We can see. So this polyploidy is seen in almost all kind of animals. So what is the significance of these chromosomal mutations and aberrations? So haploidy, polyploidy or heteroploidy produce various phenotypic changes in the organism, and deletions after phenotypic traits, they will have missing gene loci, and homozygous deletions are lethal, whereas heterozygous deletions can be sustainable. Like, and the duplications carry various phenotypic abnormalities like barred eye in Drosophila, and inversions and translocations also bring chromosomal rearrangement, but they do not produce any significant viable visible effects, and the phenotypic changes produced due to spontaneous or induced chromosomal mutations may result in new varieties of species or new species itself. Okay. So when the heritable alterations occur in very small segment of DNA, these mutations can be like only one single position or so. They are called as point mutations. Even they can be deletions, means a single nucleotide may be deleted, and insertions, new segment may be inserted in genetic level, not on chromosomal level. Then substitution, like one base is substituted with another base, so they are called transitions and transversions. Transition is nothing but a purine is um, replaced by another purine, like adenine is replaced by guanine, cytosine is replaced by thymine. That is called transition. Transversion means purine is replaced by pyrimidine, like that. So, like adenine is replaced by cytosine. Okay. So, this will affect the protein expressions. So, the gene mutations, just a second. Yeah. So, the gene mutations are very important because gene mutations are highly stable. They mutate genetically, and we can see in virus, bacteria, and man, everywhere we can see during cell division also. But this mutation is a normal process. And some genes are called mutator genes. Okay, they increase the mutation frequency of other genes. And the mutation gene may continue mutating in different directions, causing diverse effects. And some mutations is for normal origin, and some may be in the reverse mutation, and some occurrence is back mutation, which indicates a mutation that involves a chemical change in the gene, and some may affect the protein, some may not affect. And significance of gene mutation is some gene mutations are negligible and no effects. Some mutations like insertions and deletions may have major effects. They can be harmful, can cause death also, like lethal. They can cause some diseases like cancers. And in homozygous conditions, they are lethal. In heterozygous conditions, if they are recessive, they can be unexpressed. And some are deleterious, and most of them are associated with some kind of diseases. So there are induced mutations and normal spontaneous mutations. So induced mutations are induced artificially or from due to some external factors like mutagens or mutation agents, like high energy radiations like X-rays, gamma rays, ultraviolet rays. So they are induced mutations. So they were studied by Muller. Okay, artificially also scientists will give some radiation to produce some mutation, and also due to some radioactive vapors, especially in the Hiroshima after nuclear attack. So people are still, because so much of radiation, atomic bombs, still people are suffering with that mutations. Okay, that radiation is still present there. And chemical mutagens, so Robson and Orbach, so they investigated mutagenic agents, some chemicals. Okay, they like peroxide, formaldehyde, phenol, ethyl urethane, and sulfur mustards, and so on, so and so. So they can cause various mutations, and like base, they can form base analogs like purine, fibromouracil, like that. Okay, they get inserted in the DNA instead of normal nucleotides. Then temperature. If the temperature increases, they can enhance the rate of mutations. And the Muller and Ettenberg, so they have observed that in Drosophila raised at 29 degrees centigrade underwent mutations 3.2 times more than the normal temperature. Okay. So by inducing the mutations, we can study quantitative and qualitative improvement in plant and animal progeny. Okay, mainly for the study purpose. Okay, and in some like people like due to exposure, due to their work, nature of work, they may get exposed to some radiations and the mutations get accumulated. Okay, so this is the end of the topic. So tomorrow we'll continue with next topic. Thank you.